so unit so unit okay so so unit 6, right? Reproduction. Now we are going to study like 3 chapters in this unit: the sexual reproduction in flowering plants, human reproduction, and reproductive health. Now, what is the introduction given in the unit? Okay, the biology is in essence is the story of life on earth. Okay, now there is a term that is anthropogenic extinction, that is extinction because of the human activities. Reproduction becomes a vital process without which species cannot survive for long. Okay. So reproduction becomes a vital process without which species cannot survive for long. So each individual leaves its progeny by asexual or sexual means. So sexual mode of reproduction enables creation of new variants so that the survival advantage is enhanced. Now, okay, so this is the introduction, right? Now we are going to see like there is Panchanan Maheshwari, what he did. So Panchanan Maheshwari, in the botany and especially morphology, he worked in botany and especially in morphology. Okay, so Panchanan Maheshwari worked on embryological aspects and popularized the use of embryological characters in taxonomy. So the study of line and development of the embryo and all that stuff. So embryological characters in taxonomy. Okay, he established the Department of Botany, University of Delhi as an important center of research in embryology and tissue culture. Okay, so Panchanan Maheshwari also emphasized the need for initiation of work on artificial culture of immature embryos. So these days, tissue culture has become a landmark in science. His work on test tube his work on test tube fertilization and intra-ovarian pollination won worldwide acclaim. So his work on test tube fertilization, right? IVF and all that, and intra-ovarian pollination. Intra-ovarian pollination means pollination inside the ovarian, intra-ovarian pollination. He was honored with Fellowship of Royal Society of London (FRS), Indian National Science Academy, and several other institutes of excellence. Okay, so this is the Panchanan Maheshwari Gulab Jamun. Anyways, so chapter 1, sexual reproduction in flowering plants. So the context are: flower, a fascinating organ of flower is an organ of angiosperms, then pre-fertilization, that is structures and events. Pre-fertilization we are going to study in structures and events, double fertilization, that's post-fertilization structures and events, and apomixis and polyembryony, right? So this is it. Like, now plants reproduce sexually, right? The myriads of flowers that we enjoy gazing at, the scents and the perfumes that we sown over, the rich colors that attract us, are all the three as an aid to sexual reproduction, right? So the scents, the perfumes, and the the myriads of flowers and rich colors that attracts us, these are all an aid to sexual reproduction. Now flowers do not exist only for us to be used for our own selfishness. So all flowering plants, these are angiosperms. All flowering plants show sexual reproduction. Okay, so okay, now so the end products of sexual reproduction, these are the fruits and seeds. So end products of sexual reproduction, these are the fruits and seeds. Okay, so fruits and seeds. So you can see the morphology, structure, and the processes of sexual reproduction in flowering plants. Angiosperm. Now flower, a fascinating organ of angiosperm. Okay, flower, a fascinating organ of angiosperm. So human beings So okay. Okay, so the culture and farming of the flowers is known as the floriculture. Okay, so flowers are morphological and embryological marvels and the sites of sexual reproduction. Okay, okay. Now pre-fertilization structures and events. So okay, so several hormonal and structural changes are initiated which lead to the differentiation, maturation, and further development of the floral primordium. Now inflorescences are formed which bear the floral buds and then the flowers. So inflorescences are formed so after the several structural and hormonal changes in the floral primordium. Inflorescences are formed which bear the floral buds and then the flowers. Right? In the flower, the male and female reproductive structures, the androecium and gynoecium differentiate and develop. So in the flower, the male and female reproductive structures, the androecium and gynoecium differentiate and develop. So okay, the androecium, now the male reproductive structures in the flower, consist of a wall of stamens representing the male reproductive organ, and the gynoecium represents the female reproductive organ, right? So these are the pre-fertilization structures and events. Now, so the stamen, microsporangium and pollen grain. So the two parts of a typical stamen, the long and the slender stalk called the filament, and the terminal generally bilobed structure called the anther. So terminal, which is a bilobe structure, that is the anther. Now the proximal end of the filament is attached to the thalamus or the petal of the flower, either to the thalamus or to the petal of the flower, right? Now the number and length of stamens, these are variable in different species. Okay. Now a typical angiosperm anther is bilobed with each lobe having two theca. So they are dithecous. So a typical angiosperm anther is bilobed. There are two lobes, okay, with each lobe having two theca. So when each lobe having two theca, it is dithecous, right? Now often a longitudinal groove runs lengthwise separating the theca. Okay, now the theca is being separated. Okay, the bilobe nature of an anther is very distinct in transverse section of an anther. Now the anther is a four-sided tetragonal structure. These are consisting of four microsporangia located at the corners. So the anther is a four-sided tetragonal. So anther is a four-sided tetragonal structure consisting of four microsporangia located at the corners, two in each lobe. So four microsporangia located at the corners and two in each lobe, right? So okay, so macrosporangia located at corners, two in each lobe. The macrosporangia develop further and become pollen sacs. Okay, so microsporangia, they develop further and become pollen sacs. So they extend longitudinally all through the length of an anther and are packed with pollen grains. So the structure of microsporangium. Okay, a typical macrosporangia appears near circular in outline. Okay, it is generally surrounded by four wall layers: the epidermis, endothecium, middle layers, and the tapetum. So epidermis, endothecium, middle layers, and tapetum. The outer three layers perform the function of protection, right, and help in digestion of anther to release the pollen. So outer three walls like epidermis, endothecium, and middle layer. The innermost layer is the tapetum, right? Now this tapetum nourishes the developing pollen grain. Now okay, now so this tapetum, it nourishes the developing pollen grain. The cells of the tapetum possess dense cytoplasm and generally have more than one nucleus. Can you think of how tapetal cells could become binucleated? It is because there is lack of, I mean, cytokinesis. So polyploidy nature is present. Okay, so it has dense cytoplasm and generally have more than one nucleus. So when the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each macrosporangium. So when the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each sporangium. Okay, now the microsporogenesis, as the anther develops, the cells of the sporogenous tissue undergo meiotic division to form microspore tetrad. So microsporogenesis, it is formation of the microspore tetrad, right? What would be the ploidy of the cells of the tetrad? It is haploid, right? Now it's a young anther enlarged view of one microsporangium showing wall layers. A mature dehiscence anther. Okay, you can see the pollen grains are dehiscence. Now in first diagram you can see there is a connective tissue that connects, I think, the filament. Okay, then epidermis, then endothecium, then sporogenous tissue, and then tapetum, and then there is a middle layer. So epidermis, endothecium, then middle layer, then tapetum, and then sporogenous tissue. So this is the sequence. It can be asked in the exam, definitely will ask. And epidermis, endothecium, middle layer, tapetum, and sporogenous tissue, macrospore mother cells, right? So this is it. Now as each cell of the sporogenous tissue is capable of giving rise to a macrospore tetrad, so each cell giving rise to a macrospore tetrad. Now each one is a potential pollen or macrospore mother cell. Each one is a potential pollen or macrospore mother cell. The process of formation of macrospores from pollen mother cell, that is PMC. The process of formation of macrospores from a pollen mother cell, that is PMC, through meiosis is called the macrosporogenesis, right? The macrospores as are formed are generally arranged in a culture of 4 cells, that is the macrospore tetrad. As the anther mature and dehydrate, the macrospores dissociate from each other and develop into pollen grains, right? So inside each macrosporangia, several thousands of microspores or pollen grains are formed that are released with the dehiscence of anther. So macrosporogenesis, that is process of formation of macrospores from a pollen mother cell, that is PMC, are formed, and then so these are the macrospore tetrad. As the anther mature and develops, so the macrospore dissociate from each other and develop into pollen grains. So now the pollen grains represent the male gametophytes. So pollen grain, it represents the male gametophytes, right? Anther has the macrosporangia. They have the macrospore sacs, and it produces the pollen grain. So now this male gametophyte, that is the pollen grain, it has a variety of architecture, okay, of different species. Now pollen grains generally are 25 to 50 micrometers in diameter. Now they have two layered wall, that is outer is the exine made up of sporopollenin. It is the most resistant organic material known. It can withstand high temperatures and strong acids and alkali, right? No enzyme that degrades sporopollenin is so far known. Now pollen grain exine has prominent aperture called jump pores where sporopollenin is absent. Okay, so pollen grains are well preserved as fossils because of the presence of sporopollenin. The exine exhibits a fascinating array of patterns and designs. So this exine, that is outermost covering, it exhibits a fascinating array of patterns and designs. Now this exine should be hard. Okay, the function of jump pore is to produce the pollen tube. The inner wall of the pollen grain is the intine. Now it is thin continuous layer. The outer one was the discontinuous, a thin and continuous layer made up of cellulose and pectin. So inner layer of the pollen grain is made up of pectin and cellulose, right? The cytoplasm of the cell pollen grain is surrounded by a plasma membrane. The cytoplasm of pollen grain is surrounded by a plasma membrane. The cytoplasm of pollen grain is surrounded by a plasma membrane. Okay, when the pollen grain is mature, it contains two cells, that is vegetative and generative cells. The vegetative cell is bigger, has abundant food reserve, and a large irregular shaped nucleus. The generative cell is small and floats in cytoplasm. So the generative cell is small and floats in cytoplasm. So generative cell is small. So generative cell is small and floats in the cytoplasm, that is the of the vegetative cells, right? Uh, so it is spindle. So generative cell, it is spindle shaped with dense cytoplasm, and it is spindle shaped with dense cytoplasm and nucleus. So generative cell, it is spindle with dense cytoplasm and nucleus. In over 60% of the angiosperm, the pollen grains are shed at two cell stage of it, and in remaining, the generative cells divide mitotically to give rise to the two male gametes before pollen grains are shed. Pollen grains are shed to for pollination, that is three at the three cell stage. So before the pollen grains are shed for the pollination to the stigma, right? So they ultimately becomes three cell stage. So before pollen grains are shed, right? And pollen grains are shed at two cell stage in the 60% of angiosperm. So this is it now. And the pollen grains of many species cause severe allergies and bronchial infections in some people, often leading to chronic respiratory disorder, that is asthma, bronchitis. Okay. Now that the Parthenium or carrot grass that came into India as contaminated with imported wheat has begun ubiquitously in occurrence and caused the polar energy. Now pollen grains are rich in nutrients, so they are rich in nutrients, right? The pollen tablets as food supplements. In Western countries, a large number of pollen products in the form of tablets and syrups are available in the market. Now pollen consumption has been claimed to increase the performance of athletes and race horses. As you can see, these are the pollen products. They act as food supplements, right? So once they are shed, the pollen grains have to land on the stigma. So before the shade, but if they are shed, so if they are shed, the pollen grains have to be land on the stigma, right? So before they lose, so before they lose, so before they lose, so before they lose viability, if they have to bring about fertilization, okay? Before they lose viability, if they have to bring about fertilization. So there are also the viability of different. So there are also the viability, right? The period for which the pollen grains remain viable is highly variable and to some extent depends on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release. So wheat and rice, right? In some cereals such as wheat and rice, and in some members of Rosaceae, Leguminosae and Solanaceae, they maintain viability for months. So Rosaceae, Leguminosae and Solanaceae, they maintain the viability for months, right? The storing semen and sperms of many animals, including humans, are for artificial insemination. It is possible to store pollen grains of a large number of species for years in liquid nitrogen, that is -196 degrees Celsius. Such stored pollen can be used as pollen banks, similar to seed banks in crop breeding programs. So in some cereals, that is rice and wheat, in some cereals such as rice and wheat, it is for an hour, half an hour, within 30 minutes of their release. But Rosaceae, Leguminosae, Solanaceae, it is viable for months, and -196 degrees Celsius for liquid nitrogen for the storing of the sperms. And about now this diagram I'm going to cover. So scanning electron microscopes of a few pollen grains, you can see. So they have the different architectures in form, colors and shapes and all that. Typically it is 20 to 25 to 50 micrometers in diameter. Now this one is a pollen grain. It has a macrospore tetrad, right? Now it has a pollen grains. You see, it is single cell. So there are the vacuoles. Okay, then there is a nucleus. Then further stage is asymmetric spindle formation. So there is a formation of then asymmetric 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 spindle. Now there is formation of two cells, that is vegetative and generative. So there is like vegetative and generative. So vegetative, it means there is like abundant, it is bigger and has abundant reserve food materials and a large irregular shaped nucleus. You can see it is a large irregular shaped nucleus, right? Okay, now the generative cell, it is small and fruits in cytoplasm of the vegetative cell, but this generative cell is simple. It is spindle shaped with dense cytoplasm. So this generative cell has dense cytoplasm and it is spindle shaped, right? So vegetative cell and generative cell, as you can see. So enlarged view of pollen grain tetrad. So first image is a pollen grain tetrad, right? And B is the stages of microspore maturing into a pollen grain. So stages of microspore. So stages of microspore, that is. So stages of microspore maturing into a pollen grain, right?